I think as we are waiting now, we can already start with the logistics. So welcome everyone to our 31st Let's Talk About webinar session with the title of Live Imaging of Dynamic Processes Prior to the Formation of New Blood Vessels. My name is Anna Fehir and I'm a product application specialist at Confocal NL, and I will be your host today. Before we start, I would like to start with some logistics. So as you can see, we have an open chat panel on the right side of your screen. And please feel free to use this chat panel to address your questions. I will try to answer all the questions here in this chat panel, but also we will have a Q&A session after the presentations where you can ask your questions. This webinar is recorded, and after the webinar you will get a follow-up email with where you can access the recorded webinar. So if you want to rewatch it or if you want to share it with someone, then you can just freely do it. About the agenda of the today's webinar. So we will start the webinar with a presentation from Maxime Cammeraat, who will talk about the sprouting angiogenesis with the focus on the techniques of for 2D and 3D imaging of these processes. And this will be followed by a presentation and a demonstration by Soledad Ordonez, who is our new product application specialist, who will talk about the applications and the specifics of NL5, and then give a demonstration on the system and show how we can image these such processes and structures with our fast confocal system. And this will be followed by a Q&A session where you can ask your questions. So I think for now we can already start with the first presentation from Maxime. Maxime, I would like to welcome you here. Thank you for joining. And Maxime is a research technician at the Angiogenesis Laboratory from Amsterdam University. If you're ready, then I will just give it to you.
Yes, thank you for your introduction. I will just share my screen to show the presentation. So, yeah, I will give you a talk about the live imaging of the dynamic processes during sprouting angiogenesis. Yes, Anna told, I'm Maxime Cammeraat and I'm working in the group of Koert Margedant at the CCA, part of Amsterdam UMC. So I will first give a bit of an introduction on sprouting angiogenesis, which is the formation of new blood vessels out of existing ones. It's very important both for physiological processes as well as pathological processes. So healthy angiogenesis is seen in embryonic development, but also in wound healing and tissue regeneration. But also in diseases, as eye diseases, cardiovascular diseases, as well as cancer. So in response to VEGF, which is a growth factor, a tip cell branches off from the existing blood vessels and is followed by stalk cells. And they start migrating towards the VEGF gradient, and also eventually they will start forming a lumen, and then you will have a new blood vessel. This is a very dynamic process and is regulated by a pathway, which I will not go into detail about, as it is not really important for the rest of my talk. So in our lab, we have optimized the sprouting assay, which is an in vitro assay in which we can visualize sprouting angiogenesis. So to do this, we have collagen-coated beads on which we coat endothelial cells, which are UVECs. These beads are put in a fibrin gel in a 96 well plate, as you can see here. And upon stimulation with VEGF, so just normal culture medium, these cells will start sprouting. It's a very high throughput assay, and we can do very many conditions at the same time. And here in this image on the right side, you can see a sprouting assay after fixation. So this is 48 hours of sprouting and then imaged. Oh, I switched the slide. So in magenta, we see the actin, and in cyan, we see the nuclei. So this is confocal imaging of a fixed sprouting assay. The sprouting is a very a three-dimensional process, as you can see also in this video. The complete depth of this whole bead, including sprouts, is about 250 micrometers, so that is quite big. And in the image on the left side, you can see the depth code, in which the different colors represent the Z position in the gel. So on this side, this is closer to the objective, and then as it goes to the red color on the other side, it's further away from the objective. So to analyze our sprouting assays, we have a macro in image J, and here we see the image that is acquired, and then the macro can determine the bead versus the sprouts, the number of sprouts, the sprout length, the total network length of all the sprouts, as well as the number of cells. But all of this that is done now on fixed material, and as I told, the sprouting angiogenesis is a very dynamic process, and we want to see fast changes, and also like such as like the elongation of sprouts, but also the retraction of sprouts. And this is something that we cannot see once it's already fixed and in the end point of the experiment. This actually comes with quite some challenges, because repeated exposure over time results in photobleaching, where the staining intensity becomes less. And also the big Z range and the short time intervals to capture the dynamics makes this even harder. This often results also in phototoxicity and cell death, which is what we don't want. Also, to visualize this process, we would need expression of fluorescent proteins, which can be achieved by lentiviral transduction. But this can also be a problem, as UVECs have a quite short lifespan, as they are primary cells. So yeah, it would be best to have a quick way to label them and visualize them, which we can do with dyes. But conventional dyes as Heugst and Phalloidin are often very toxic and also better for fixed cells. So we want to develop approaches for live labeling and imaging by time-lapse microscopy, but also we want to be able to combine this with RNA interference, such as knockdowns of specific proteins, so we can compare different situations and different in different cells, or well, so both UVECs, but yeah. So for this purpose, we use the spirochrome probes. These are non-toxic, and they exist in equilibrium between the on and off state. And the big advantage of these probes is that they are only fluorescent when they are bound to their target protein, which is in this case actin. And this is very convenient because it means that the probe can stay in the medium during imaging, and there's no need for washing it away. This also makes the signal very consistent during imaging. So we have tested these probes in our sprouting assay, and on the left we can see the actin that we have labeled with the probe, during live. So this was before the sprouting, and during the sprouting. So this is live labeled cells, and on the left, or the right, I should say the right, we can have the same beads with the same sprouts, but stained after fixation with a phalloidin stain, which is a conventional, widely used stain for actin as well. And as you can see, there is no changes in structure, and also there's no impairment in sprouting. As you can see, there's very nice sprouting, and it's not. It's just as efficient as without the probe. Another thing that's very convenient is that these probes can also be combined with antibody stainings. So on the left, we have cells live labeled with actin probe, and on the right, after fixation we stained this again with the phalloidin and DAPI for the nuclei, and anti-phosphotyrosine staining. So this in green, the phosphotyrosine is visualized. So this is the dots that are actually focal adhesions, where the cells attach to the matrix, and you can often see this in the end of stress fibers of actin. So this is long stripes that you can see here. But this is still a fixed image. So we also have a workflow for live image in both a 3D setting and a 2D setting. So in a 3D setting, we use our sprouting assay and just add the probes in the medium. And in a 2D setting, we can grow a confluent monolayer, Vuvex, and also add the probes in the medium, and then start imaging. So here I have some 3D and 2D videos of some experiments that we have performed using these probes. So the first video is a sprouting assay, and we've zoomed in on a tip cell. And this is quite still long time interval of about 20 minutes, but you can see there's a lot happening during this period. And as again, the nuclei is in cyan and the actin is in magenta. And the same is here in the same stains are here in the 2D setting, where we have Vuvex plated, and then in the middle here we have made a scratch. So it's just basically a wound in the monolayer, and you can see them migrating to close the scratch, to close the hole on the plate. And in both ways you can see that the probes are very well retained, and the signal stays more or less the same during imaging. So we would also really want to combine this live imaging with RNA interference via lentiviral transduction. So to produce these lentiviral-like particles, we add a short hairpin RNA and helper plasmids to packaging cells, the HEC293T cells, and after 24 hours we can harvest the medium, which contains the viral particles. We do this at 2 days and then pull the harvest and filter it. And then we can transduce our Vuvex. So it's, we just add the viral particles to the medium. And after 12 hours, we can select for the positive cells. So there's two ways to approach this. So the first thing that we can do is knock down a specific protein, and then afterwards we can select for the specific the positive cells using antibiotics. But it's also possible to do, instead of a knockdown, make a fluorescent protein to overexpression, and then there's no need to select with antibiotics, as you can just select the cells by eye using the fluorescence. So I've also the examples of this So at first I have UVECs that are transfused with the GFP vinculin virus. As you can see, the cell edges is also focal adhesion marker, but it is mainly here on the cell edges that's visible. And this can be combined with the probes and with the live imaging, and we can follow this over time. And the other RNA interference that we can do is a knockdown of a specific protein. And in this case, we have performed a control experiment where we use a scrambled sequence, so it's not a knockdown of a specific protein, but it undergoes the same procedures as it would if it were to have a specific knockdown. And as you can see, this is still very much possible as the labeling is very fast, and then we can combine it with the transductions that take a few days. So to combine, to summarize, all of that we have is that we can do fast and bright labeling of the different cell compartments, such as the nuclei and actin filaments. And this is suitable for both live and fixed samples, but most importantly, it allows time-lapse imaging for actually quite some time in both 2-dimensional setups and 3-dimensional setups. And at last, I want to thank my group. So Kurt Margoland, who is my group leader, Wendy Stam, Gabriella Holtz, Dimitrios Svakianakis, and all the other members of the angiogenesis laboratory. And then this is where I would like to end the presentation, and I will also be happy to answer any questions.
Thank you very much, Maxime. Thank you for this interesting presentation. I don't see any questions at the moment, but I was wondering, I saw, and you mentioned also that you can image these processes for quite long. And how long do you usually image them, and what are the limitations there?
So we have managed to image like in a 2D setting for about 15 hours, which is already quite long because we expose the cells every 10 minutes. So that's, well, I cannot do the math right now, but it's quite some time. For a 3D setup, this is a bit harder because, of course, the Z range that we want to capture is bigger, so that also means more exposure. But at least we now managed to get around 8 hours of footage, which is quite long. It shows some nice dynamics. So.
Great, thank you very much. Then I think in the end, if we will get some questions, then we will get back to you, and thank you for the presentation again.
Yes, you're welcome.
And now we will continue with the presentation and the demonstration with Soledad Ordonez. With the last minute technical issue, we cannot do a live demonstration, but we got some nice fixed samples from Maxime. So Soledad will use those samples to show the performance of NL5.
Thank you, Anna, for the introduction. I'm going to quickly open my presentation. I hope you're seeing it now. A little summary first of what I'm going to talk today is first I will focus on NL5, which is one of our products, the specifications of it, the applications, and with that I will share with you a few movies. And finally I will, with some fixed samples that Maxime has kindly given to us, I will do a live demo so you can see how our microscope works. So this is NL5. It is a line scanning confocal microscope. What you see in the picture, the orange box, is the scanning unit, and behind it there is a camera for detection. You can see it's very compact. It can be connected to either the left or the right side of the microscope, and it can be an inverted or as well as a upright microscope. So how it works? It actually has a speed of 25 up to 25 frames per second, and it can use very low laser power. It can reach a resolution of 2K by 2K, and it also allows fast color switching. But now we can also say that you can do a deconvolution of your images during image acquisition, because we have combined the software of microevolution with our unit. So this is possible also. For applications, I want to start first with a little bit of maybe cell dynamics. This is very nice because with a low phototoxicity you can, and the speed of NL5, you can capture through long time periods very fast dynamics inside the cell. This could also help in immunology, for example, cell signaling, infection models, and 3D cell visualization. So here I'm sharing with you a movie of HeLa cells, and they are the cytosol is with a fluorophore GFP, and you can see now we have done a set stack. I hope the movie works in time, and you will see how even though we have captured so many images of the cell, the cell looks happy, and you can see, I will repeat it once more, the filaments moving. So the set stack through time, one stack per 10 minutes. Another application could be also for developmental biology, when you use model organisms. And this is a movie that I share about zebrafish embryo. And you can see the endothelial cells that have been labeled with GFP, and how they move while the embryo is developing. This is 1-day imaging, and we have done set stacks continuously every 10 minutes. So 500 in each set stack is 500 slices, and you can see that the embryo is really not affected by phototoxicity. You can even see the development of the heart, and how cells migrate. It's a very nice video. Another application can be high throughput screening with multicolor imaging. So this could be also helpful for drug discovery, for example, and plant breeding. Here I'm showing you a movie of HUVEC cells, and in 3 colors. So you can see the nuclei in blue, actin in yellow, and microtubulin in red or magenta. This is also a set stack in time, and you really can see that the mitosis is not affected by phototoxicity. To finalize, I really want to show you how the microscope compares to another fast confocal option that it is commonly used and very common in the market, which is a spinning disc. So this application is deep imaging, and I'm showing you here an algae volvox, and it's a very thick sample. So we can go to, well, now I will go to 90 micrometers. This is 20 micrometers, so it's really on top of the cover slip. And here the difference is not so evident, but you already can see the intensity of the laser used by NL5 compared to spinning disc, which is much lower. Sorry, I think here the nanowatts is not right. In here at 60 micrometers, it is the right nanowatts. Yeah, if you go deeper to 60 micrometers, you really can see the consistency of the image and the nice noise to light ratio. You can see that the brightness to noise ratio is very nice in NL5, that you can have with very low laser power a really sharp image. And if we go even deeper, you can really see how the crosstalk between the pinholes of the spinning disk really deteriorate the quality of the image, while for NL5 the resolution stays the same, the quality of the image. So that was for my presentation, and now I will go for the live demo. So I hope you can see now my screen. I'm going to use for this demo a Micromanager, which is free software. I hope it's big enough for everybody to see. So a little bit about the user interface. You have on the left side buttons to acquire the image, and on the right side you have controllers for the confocal units, the lasers. So first let's look at the bright field. I am opening the settings, and I'm changing it the view to grayscale. Here you can see one of the beads that Maxime was talking about, and the cells that are sprouting. And I'm moving a little bit on the set. So we are in focus. We can go to the halogen lamp and see maybe. Also, we are not on the set. Yeah, here are the nuclei of the cells. I'm going up on the set. Yeah. So let's see here. I will change the color to maybe blue. Okay, and I will stop live imaging. I will do multi-acquisition for you to check differences between using LED lamps and the confocal unit. Here in channels, I will specify first our fluorophores, our lasers, sorry, our LED lights, and acquire. Let's zoom out and yeah. So I'm just getting a good threshold, and I will leave this one here. I hope I'm not confusing you. And I will immediately acquire with the confocal unit. Let me see. Yeah. So you can see here the comparison between the bright field. Maybe this one I will open again, image inspector, and put the same colors so we can. Let's see. Oh, I did it the other way around, sorry. Yeah. Now I will show you how to do a set acquisition. I'm closing a few windows. Yeah, so we are live now. And I will set the boundaries of the set stack. The upper one and the bottom. So now you will see that the number of slices are 27. This is good to show you, so you can see the speed of the microscope. Sorry for the movement. We can use 2 time points. I think that's more than enough. Can put even less. 10. Yes, and we can acquire. Now let me go back to settings. Yes, so you can really see now how fast the imaging was and how nice the contrast is. And the low laser power that you need. Well, thank you. I think Anna now can.
Thank you, Soledad. Could you let us know what was the speed you were currently using for this experiment?
Oh, yes. Let me see. I didn't. Pay attention. Oh, I was working at 5 frames per second, but you can put it up to 25. Yes.
Okay. And then you were you compared the wide field image with the confocal one
Yes.
for the beginning. Okay. Then thank you for the presentation and also for the demonstration. I don't see any questions, but if you have any questions in mind, please feel free to address it to the sales@confocal.nl, and we will forward the questions to Maxime and Soledad. Then you will get also a follow-up email with the recorded webinar. And you can find also more information of our systems on our website, and you can follow us on social media. You can also book your own demonstration, and we will follow you up. If you want to see some more things, you can also image your samples as well if you send it to us. So you can find all this information on our website. So I would like to thank you for Maxime and Soledad, and also for the attendees. And I hope you really enjoyed it as much as we did. And hope to see you on our next webinar. Bye.